Namaste, we welcome you all to Sakkath Bengaluru vlogs. Today we are at a very unique place, and this place is Thomas Biotech and Cytobacts Centre for Biosciences. This is a place where papaya plant is grown through a unique method called the tissue culture method. So there is no normal plantation method. So this is a different and unique method where we can witness the entire process of how papaya is grown through the tissue culture method. Gayathri, what do you feel about this?
That's what Sindhu, for papaya, wherever you go, they are charging us more. Why? Because the nutritional value, what doctors are telling or dietitian are telling. So even I prefer having papaya at the breakfast time. So today let us help the farmers how we can grow papaya through tissue culture. Let us know about that process.
So come, let us witness the entire process of how papaya is grown through the tissue culture method. Namaste. Today we have with us Dr.Pious Thomas, CEO and Director of Thomas Biotech and Cytobacts Centre for Biosciences. Sir, we extend a very warm welcome to Sakkath Bengaluru vlogs. Can our viewers know something more about yourself, sir?
Yes, I come with a background in research. I worked with Indian Institute of Horticulture Research as a scientist for almost 3 decades. I opted for VRS as a principal scientist in 2018 with a view to take my research findings for the benefit of the farmers. In 2019, I launched this startup. Here we work mainly on crops like papaya and pomegranate and also acid lime. Our priority is to give quality planting material to the farmers and also to give further support to them.
Sir, here we see so many awards and recognitions that you have received, and one of these photos you are also receiving some award from our former Prime Minister Manmohan Singh. So can you tell our viewers more about this?
Yes, I was fortunate to receive several national awards. The one which I received from the former Prime Minister Manmohan Singh was Harikrishna Shastri Memorial Award from Indian Agriculture Research Institute. This was for my research contributions to the field of horticulture biotechnology. I was also recipient of national awards, DBT Overseas Association Award, BP Pal Memorial Award, and also last year Biotech Enterprises Award from the Karnataka state government.
Sir, this is, you are telling that tissue culture incubation room.
Yes.
So can you explain the procedure, sir, how it goes on?
Yeah. These are all the nutrient enriched media in which the tissue culture plants are grown.
Okay, sir.
This contains all the nutrition which are required for the plants, what is available for the from the soil, and additional material because light is not there, so we provide all the nutrients required to supplement again with the light. So this is to be done under totally aseptic conditions, that is free from all microbes. So these formulations we standardize, and then once the formulation is optimized, then the media is prepared, that is to be sterilized, and all the operations are done under aseptic conditions.
Okay.
This is called laminar airflow. So all the culture transfer work is being undertaken in sterilized.
Okay, sir. So these are the sterilized bottles
Sterilized
you
bottles.
told. And the medium. I'll ask you one question, sir. So most of the people, they are not aware of what is tissue culture.
Yeah.
So you told about that medium, and I could see some few small plants.
Yeah.
So how do you start with?
Here we go for rapid multiplication. Initially, we have to sterilize the plants and then establish the cultures. And this is the initial stage of establishing the cultures.
Sir, what is that smaller plant? Where do you get from?
This is from the shoot tip of the plant. Usually we
Shoot
take
tip of the papaya,
papaya
plant. Okay.
and from which it is initiated, and then gradually it is taken through the.
So when you put them in these test tubes,
Yeah.
what is the size of that shoot
It will
tip,
be
sir?
very small, just about 1 centimeter.
1 centimeter.
Yeah.
Okay. So the first stage is you will put them in the medium of.
Yeah, this is to be initiated with.
Okay.
And once the culture is established, we go through the propagation cycle.
Okay.
Here, this is the proliferation. Here, you could
So
see the
this plant is transferred from test tube to the bottle.
bottle.
Okay.
And then from this bottle.
Is it the same medium what you have put in the test tube as well as in this bottle, sir?
Slight modifications are there.
Slight modification.
Yeah, so this is
Okay.
that is to help the initial establishment. This is for the proliferation purpose. And from this one, from each plant, each bottle, usually we'll have about 4 plants per bottle.
Mhm.
And after a period of about 3 to 4 weeks, each plant will be producing about 3 to 4 cuttings we can make from that one.
Okay.
And then it can be transferred to sort of cuttings from one bottle can be transferred to 4 bottles.
Oh.
So that way, after another 3-4
So
weeks, it will
there
be
will be
transferred.
multiplication of
So that is
plants.
what is happening is about 4 into 4
Okay.
into 4. So that way,
Mhm.
over a period of time, if you go for theoretically, we can reach about million plants. So that's a very fast method of propagation, multiplication. So only thing, all these are to be done under very careful aseptic conditions. And
Okay.
we require, a lot of optimization required, particularly for papaya. To my knowledge, papaya tissue culture is not done successfully commercial scale by anybody in the country. And this is some, we are demonstrating here practically.
Is there any standard of maintaining temperature and all
Exactly.
to
Yes.
do this, sir?
Yes,
Yeah.
we are required to maintain very ideal temperature conditions. So we usually
Okay.
maintain about 25 to 26 degrees
Okay.
centigrade here. And light is provided for up to 16 hours a day here.
Okay, like this, sir. Is that laminar airflow used for, sir?
This is giving a filtered air.
Okay.
So all the operations are done under the filtered air in this. So you'll be getting a sterile air. So all
Sir, I'm seeing this gas-fired autoclave. What is this all about, sir?
I mentioned about the total aseptic conditions required for the tissue culture work.
Okay.
All the media bottles whatever prepared, they have to be sterilized.
Okay. We just saw the plants in the bottles down. So how about this stage, sir? Have you transferred it directly from the bottle to the?
Yeah, this stage is called hardening or the acclimatization phase.
Okay.
The plants in the bottle are very delicate. They are really tissue culture babies.
Mm-hmm.
So to make them withstand the outside conditions, they have to be brought very gradually to the to the outside conditions.
Okay.
So I would say this would be the
Second
this is something
stage.
called the second
Yeah.
stage, and this is the third phase.
Okay.
This would be the fourth phase.
Mm-hmm.
These are almost
Ready for.
These are the plants ready for supply to the farmers.
Okay.
The polyhouse you are seeing the
Yeah,
plants
yeah.
which are really
Yeah.
ready for.
So once
transfer.
the plants are ready, you just transfer them to the polyhouse.
Yeah.
So is it a same plant or different one, sir?
These are very specially treated plants.
Okay.
We sell the plants or supply the plants all over the country through the Amazon nursery.
Okay.
So here the plants would be packed individually in boxes and sent across. So sometimes it may be reaching the destination in 3 days, 5 days,
5 days,
sometimes
okay.
up to 10 days. And we have got the plants to withstand such long distance
Packing
transportation.
and how is the response on Amazon, sir, for the saplings and all?
Amazon response is very good.
Okay.
We have been getting good demand and
Okay.
also good reviews about the product.
Okay. So Dawn Delight, is it a name of the breed or how is it, sir?
Yeah, this is something a new tissue culture papaya line which we selected,
Mm-hmm.
and this information is also published in the International Journal of hot size.
Okay.
Highly
This
seeded.
Thomas Delight is also name of the breed, sir.
This is new, yes.
Okay.
These are only hermaphrodite plants.
So
That
all
means all
these
the
are
plants-
of tissue culture.
Yeah, these are all tissue
Okay.
culture, yes. Dawn Delight has got smaller fruits, all
Okay.
female plants,
Okay.
and it has got the potential to give seedless fruits because it can set the.
So is there female plant and male plant separate? How is it, sir?
Yeah, papaya originates was called dioecious, that is separate male and female plants.
Okay, sir.
But nowadays we have got newer varieties where we
Okay.
have got gyno-dioecious type. That means the population would have a combination of female plants and hermaphrodite plants. This is the type of fruits coming from Dawn Delight.
Dawn Delight. That is the female breed,
This is the
sir?
female breed.
Okay.
And this is like about half kg up to 1 kg usually you get the fruit size.
Okay.
And this is a small fruit from Thomas Delight.
That is male breed,
This
sir?
is hermaphrodite.
Hermaphrodite.
Male plants will not be having the fruit production capability. They only serve
For
as
the
the source of
pollination.
pollination. And the
So how about the taste of this round papaya and this
yeah,
longer one, sir?
there is not much difference in the taste, but
Okay.
there is a difference in the texture of the pulp and the thickness of the pulp. For instance, if I cut this,
Oh,
you can
it's a
see
seedless.
it's a seedless material.
Okay.
The pulp thickness is not that good, but it's quite firm.
Okay.
And even if you cut the fruit, you see here there is no seeds at all, so we don't require to remove the
Remove
seeds.
that part.
So,
Okay.
unlike that one in this hermaphrodite material, if serving seeds, usually there may be fully packed with the seeds. Here it is relatively less, and you require to remove the seeds for the
Yeah.
for consumption. So if it is fully packed and if it is fully ripe, then the surface would be disturbed, and such fruits, cut fruits, after half an hour, I don't think it will be fit for.
Fit, okay.
well,
So
well,
you mean to
consumption.
say this round fruit
The
can
seedless-
stay for long
the-
compared to the other one?
Yes, the seedless fruits are slow in ripening,
Okay.
and once it is cut, we make the segments or the pieces and keep. It can remain
it
fresh,
can fresh for longer
24
time.
hours to
Okay.
48 hours if,
Okay,
if you are
okay,
keeping in a closed container under refrigerated condition.
So when you make this tissue culture thing, sir, are you doing it for both the breeds or?
Yeah, we were doing for both the lines.
Okay.
And what we found is that there is
Much demand for the
smaller
seedless
size
one,
fruit, seedless and smaller
round
size
fruits.
People who would not like papaya at all because it's appeared as a very soft fruit.
Soft, yeah.
But this one is preferred, I mean, liked by many people who
People.
do not usually taste papaya.
Got it, sir. Got it. Thank you, sir. Hello, sir. Sir, we have come inside the net house. So, what is the use of this net house for the papaya plant, sir?
This is an insect-proof area
Okay.
to protect the plants from vectors of virus disease.
Okay.
Usually aphids cause the virus disease in papaya. I mean, they spread the virus disease in papaya. Either we go for this net cages.
Okay. So, you do sell the net cages also.
Yeah,
This is for the
is net
individual
cages.
plants, sir?
This can be used for the individual plants in the initial stage.
Okay.
Up to 4 months it will be helpful.
Okay. So, either they need to use the individual plant net or the entire net house. What is the maximum order one farmer can give, sir? Like number of saplings? How many can you supply as of now for the one order?
We supply up to 5,000 plants per order now.
Okay.
And our annual production capability is now about 1 lakh to 2 lakhs. What we are aiming is now, this is to transport the plants from here to distant places is also a difficult thing. So what we are aiming is to give the technology to different companies or individuals or FPOs or NGOs who are disinterested. Coupled with the, along with the technology, we are also planning to give them the training for the staff.
Sir, namaste and welcome to Sakkath Bengaluru Vlogs YouTube channel. So this is a unique method of growing papaya.
Yes.
So through the tissue culture method.
Yes.
So can you please tell our viewers how this method is, how do you use this method to grow papayas?
Yeah. Normally papaya is propagated through seeds.
Okay.
But in this tissue culture, we take the meristematic region, the tip of the plant, and establish the culture. And from there, it is further multiplied through the cloning technique, which is called tissue culture.
Yes, sir.
So we have all the plants like this growing in tissue culture medium.
Okay.
This contains all nutrients which are required for the plant growth, which we work based on the research.
Okay.
Do you differentiate between the plants which we grow through the seeds and plants which we grow through the tissue culture?
Yeah,
What is the difference?
papaya has got different sex forms, like
Okay.
male plants, female plants, and hermaphrodite plants.
Okay.
And when you go through the seeds, it is not. We are not sure whether it is going to be which type of plant, whether it's going to be female or male. But now we have got this methodology where we take, suppose we are taking the plant from the female, it would be all female plants through this cloning. If it is hermaphrodite plants, it would be all hermaphrodite.
Okay.
And there is no need of male plants in papaya as of now because of this special condition of this particular variety.
Okay. So, but why tissue culture method? Because when the plant is grown naturally, so why should somebody opt for the tissue culture process?
Yeah, tissue culture has got very specific advantage that if you have got any elite material and a particular sex form.
Okay.
So for instance, we have got two varieties. One is called Dawn Delight. It is of only female plants. If you are going through the seed propagation, you will be getting a mixture of female and hermaphrodite plants, which would have different size of fruits, type of fruits, size, weight, and everything.
Okay.
But when you have this tissue culture, main advantage is that you can get one uniform type of plants. For instance, female here in Dawn Delight, or hermaphrodite plants in Thomas Delight material.
Okay.
Sir, you are doing it to help to the farmers.
Yes.
So, for which breed you have the more demand? Like you told about two types of breed just now.
Yeah.
So, for which breed there is a huge demand in the market, sir?
Dawn Delight is the one which is only female plants,
Okay.
and it does got almost medium sized fruits of about 3/4 kg to 1 kg. Or 500 gram to 1 kg. That range is the ideal one for the nuclear families this time.
Okay.
So that has got great demand, and also added to that it is seedless type,
Okay.
and that has got great advantage in terms of keeping quality and the consumer acceptance of the fruit.
Okay. So, how do you want to, give these products to our viewers? Like, if any farmer want to take up this technology or any entrepreneur want to take it up on their own.
Yeah.
So, what are the suggestions that you would give to them, and how should they reach out to you?
We are confined to our service in Bangalore, but to reach larger area all over the country, we are now opening up this technology to the users in different parts.
Okay.
So any company or FPOs or NGOs are interested, or individuals are interested, they can buy the license the technology and then start the program.
So, they can approach you anytime.
Exactly.
Okay.
So we are planning now to give the technology, and simply giving the technology may not be helpful. So we are planning to give training for 2-3 staff maybe from their company for a period of 1 to 2 months, and teach them everything part of it. And then if they are interested to take this same culture, that also can be given to
That's
them
very good,
on a what I call royalty basis or payment basis.
And how about the duration, sir? In case if we propagate through seeds or tissue culture, is the time remain the same?
Yeah, it's more or less the same.
Okay.
Sometimes we find a bit of early flowering and fruiting in tissue culture plants, because they would be going through a longer duration of hardening.
Okay.
So that compensates, and so usually we get flowering in about 3 to 3.5 months time, and then fruiting in about 7 to 8 months time.
So it's almost the same.
Yes.
Okay.
So,
Or
is
even
it
like
more
better sometimes.
expensive for the farmers
Yeah.
to cultivate through tissue culture?
No, to cultivate through tissue culture would not be, that would be equal to the normal plants, but
Okay.
the cost of tissue culture plants are a bit on the higher side,
Okay.
because it takes almost 6 months. We require a lot of investment and technological know-how to do this one, which makes it more expensive.
Okay.
But what I feel is that the farmers would be very much compensated or very much rewarded
Okay.
with the tissue culture plants. Why? Because they get the desired. For instance, Dawn Delight papaya gives about half kg to 1 kg medium sized fruit, which are very much in demand.
Okay.
Those fruits are easily sold in the market, great demand, and also that fetches better premium price in the market. So a farmer invests, say, almost 5 to 6 lakhs per acre,
Okay.
including, they generally say about 60,000, 70,000 for per acre. But if they take into account the investment which they put for the labor and other things over a period of
All put together.
it almost 5 to 6 lakhs. So the cost of the planting material becomes a very small share. So the quality or good quality planting material forms the basic foundation for the good produce. So even if the farmers say puts about 20-30 rupees extra for the plant, when it comes to the returns, it will be going in lakhs.
Yeah.
So that is the real benefit through the tissue culture.
We have seen lot of saplings outside. How do you market them, sir?
Usually we take part in the Krishi Melas. We also publicize through the social media.
Okay.
And more than that, the farmer to farmer contact. They come to know about this
Word
product.
of mouth.
Word of mouth. And they contact us, and usually that is the best way to reach the farmers. And we are supplying whatever plants are available. We are usually marketing through that one. And we also market through Amazon throughout the country.
So what about the scale of the orders? Like, do you get, so is it medium, small scale? How is it, sir?
We get small to medium scale orders now.
Okay.
So usually about 1,000 to 4,000, 5,000 plants is the one which we generally follow.
So any plans to expand that capacity, sir?
Right now, not here. We are planning to tie up with other agencies or interested people to expand the production all through the country.
Okay. Finally, what do you like to tell our weavers and farmers, sir?
Yeah, we would encourage farmers to take up papaya cultivation because this is a very healthy, common man's food.
Yeah.
I don't think any other food is available in the market at such low rates, at a very nutritious medicinal fruit. And the farmers, if they are interested in growing, we give the quality planting material. As I mentioned earlier, we are trying to propagate this material, the technology all through the country through the interested people. And we also like to give more returns to the farmers. See, for instance, we get the good quality fruits. They can go to the niche market or can go even to the export market if there is a group of farmers producing large scale, and then they could form an association to export the fruits. And of late we have also come out with a processed product from papaya. Okay. One to launch is papaya honey jam. It's a very special product. I mean, usually papaya based products, I don't think many products are available in the market. It's a very unique product. We are making it available here and also all over the country through the Amazon grocery store. So anybody interested, they can just Google search for papaya honey jam in Amazon. They will be able to get this product and then see the benefits. And for the farmers, they can contact us here for the requirement for small quantity to large numbers, which within our current production capability we are supplying. And the number to reach us is 8095244466, or they can write the email to thomasbiotech.opc@gmail.com. And our location is at Amruthahalli on the Bengaluru Airport Road near between Hebbal and Yelahanka. And if just Google for Thomas Biotech Amruthahalli, it will take you to the correct location.
So thank you very much, sir.
Thank
Wish
you.
you all the best.
Thank you.